Good morning, guys. Happy Thursday. It's day 6,000 of quarantine and self-isolation. Today has already been a day, so I appreciate your patience with me and lecture today. Today's mug is "You Had Me at Meow." And there's just not enough coffee in the world for today. Anyway, we're going to start getting into the meat and potatoes, so to speak, of the urinary system, and we are going to start discussing some of the more direct physiological activities that are happening in the process of urine formation. So as we go through this, I'm going to kind of jump around a little bit. So make sure that you're paying attention to the slide numbers on the side here. And, you're going to keep going back and forth between different slides as you're studying anyway. So you might want to just make a little asterisk or, circle that slide or something like that to continue to go back for referencing. So this is one of the slides that we kind of skipped over yesterday, Tuesday. It's like, are your days starting to blur together too? Because if it wasn't for my alarm telling me that I had stuff to do on a daily basis, I'd have no idea what's happening. Tuesday, we skipped over the slide and a couple other ones, and the reason is that I think it's important to, discuss the different aspects of urine formation in terms of how they apply to the different segments of the nephron. Okay, so here we have our basic overview of urine production, and when we produce urine, we are going to go through a series of steps, and I say series, but sometimes some of the steps are occurring at the same time. But there's basically three things that are happening that need to happen throughout this process of urine production. Okay, so we already kind of discussed one of them, and that is the filtration aspect. So filtration, we said, is the process of forming urine. The initial filtrate, which is going to become urine, we form that by filtering blood through this filtration membrane that's made up of our capillary and our podocytes. And as we filter that blood, we create a filtrate, which is very similar to that interstitial fluid that we have leaving from our tissue capillaries and going out to bathe our tissues. The difference is that because we have structural differences in our capillaries as well as the membrane here, the components of that filtrate are going to be different than what you would have with interstitial fluid. So for example, with our filtrate, because we have fenestrated capillaries and we have these filtration slits that are within these little projections or processes of those podocytes, we're going to allow larger molecules to be able to pass through those compared to what we would have being able to pass through a continuous capillary and form interstitial fluid. Okay, so we're going to have a lot more solutes and larger substances that can cross this membrane and create our filtrate. So going back here to filtration, filtration is that movement of materials across that filtration membrane into the Bowman's capsule to form filtrate. That is the initial stage of urine production. Then we have two other activities that are going to take place. We have tubular reabsorption. So keep in mind that because we are losing so much fluid out of the blood, if we don't reabsorb a lot of that fluid, and especially some of the important solutes, then we would produce massive amounts of urine, and we wouldn't be able to maintain a blood pressure because we're losing so much of our blood volume in this filtration process. So tubular reabsorption is very, very important. One, to reabsorb water, and two, to make sure that we're reabsorbing nutrients and solutes and things like that that our body still needs. So that purple arrow right here is showing you basically what is going on with reabsorption. We are taking substances out of the filtrate and putting them back into blood circulation. So solutes are represented by the purple arrow. The orange arrow is representing water. Okay, so both of those types of things are going to be reabsorbed. And then tubular secretion is something that we do at the very end of our process of urine formation. And that's because anything that we want to get rid of and have make sure that it's passed out in the urine in its final form, we don't want to put those things in at the beginning because there's a chance that they could be reabsorbed at the beginning of the nephron. So all of our tubular secretion is going to be in the distal part of the nephron, and that is going to be taking additional substances from the blood and placing those into the filtrate so that they can be disposed of. Okay, so these are our overall steps associated with urine production, and our different segments of our nephron are going to have different aspects of this urine production associated with them. So if we skip back up to this slide, which I said we would talk about today, this is going over the histology of the nephron. And this is another slide that you're going to want to go back and reference as we continue talking about the different segments. So here we can see different representations of the types of cells that you would see in the different parts of the nephron. So we already talked about the Bowman's capsule, and we talked about the fact that that filtration membrane and our layers of our Bowman's capsule, as well as our glomerulus, are going to be composed of simple squamous epithelia. Okay, so our capillary endothelium, our podocytes, and that parietal layer of the Bowman's capsule are all simple squamous. Now, in particular, we said that the simple squamous epithelia of the capillary and the podocytes are important because of that function of filtration that is occurring at that first part of our nephron. So as we continue down through the rest of the tubes of the nephron, we're going to see different cell types depending on the function that's going on in that area. Okay, so in our proximal tubule, we're going to have simple cuboidal. And do you guys remember what cuboidal cells are associated with? Cuboidal cells are typically going to be associated with types of secretions. Remember, they're going to line all of our ducting systems. So, for example, your common bile duct or your pancreatic duct, your hepatic ducts, all of those type of ducting systems, as well as your salivary gland ducts, all are going to be lined with simple cuboidal. Okay, so that's a standard in a tubing system. But we have a specialized feature on these cuboidal cells, and this specialized feature is microvilli. Okay, so if we typically think of simple cuboidal associated with secretion, but now we have microvilli on these cells, that gives it a whole nother functionality. So microvilli we know are going to be important for absorption. So in this area of the proximal convoluted tubule, by having these microvilli, we increase that surface area, making this a prime area for reabsorption of different things. Okay. As we transition down into our loop of Henle, we divide our loop of Henle into a descending limb and an ascending limb, and then you have the tip of the loop at the bottom. So as we descend, okay, we're going to transition from a cuboidal epithelium, like we saw in the proximal convoluted tubule. We're going to transition from that, excuse me, into a squamous epithelium, okay, so simple squamous. So let's think about that. If we are transitioning from cuboidal to squamous, how do you think that that's going to affect the movement of things? Do you think it's going to change what we're reabsorbing? It will. So as we transition from cuboidal to squamous, we are going to change from absorbing lots of solutes to absorbing lots of water. okay? And then as we ascend, that ascending limb, we're going to transition from that squamous to cuboidal. And then in our distal convoluted tubule, okay, we return to that basic cuboidal epithelium, but we have very few microvilli, okay, which means that we're not going to be doing a whole lot of reabsorption here. But guess what? Would we say that our simple cuboidal we're typically associated with? Secretion, okay? So in our distal convoluted tubule, this is where we're going to be doing a lot of that secretory activity we saw is the third step of urine production. And then in our collecting ducts, like we said, our ducting systems typically associated with cuboidal cells, okay? We're not doing a whole lot of exchange in the collecting ducts. So this simple cuboidal is just going to allow for this movement of fluid from our nephron to our papillary duct. All right, so that is getting us set up for talking about the different segments of the nephron and what they do. You're not going crazy. The video did stop. I needed to address something, and now I'm back. So, again, we have our 3 stages of urine production. And we're going to go through each type of, each stage of urine production individually. Okay, so we're going to start with filtration. And again, I want you to visualize that Bowman's capsule, that renal corpuscle, including our Bowman's capsule and our glomerulus, as well as the filtration membrane that's allowing for this filtration to happen. Okay, so some key words here that you want to be familiar with: filtrate. Okay, so filtrate is the product of that filtration. Okay, so what it's going to contain is mostly water, small molecules and ions that can pass through that filtration membrane. So it's going to pass through those fenestrae, and it's going to pass through those filtration slits between the podocytes. What is driving our process of filtration is differences in pressure. Okay, this is very similar to things that we've talked about before in multiple systems, but if it helps you to visualize something that you maybe want to review for your final exam, go back and look at what we talked about with the movement of fluid across the capillary. Okay, it's the exact same type of thing, except now we're moving more fluid because we have those fenestrae and we have those capillaries, and those filtration slits. So let's bold this, make it red, and underline it, okay? Because pressure differences are what is allowing for that filtrate to be formed and for fluid to move across that filtration membrane, okay? That might be important, maybe. So the next set of things that are on this slide, I'll go over them quickly. You don't need to know them in detail. Renal fraction, this is the total amount of your cardiac output. Okay, so if you look again back to the first exam material with the cardiovascular system. Oh my gosh, can you guys hear my chicks in the background? They're freaking out. Cardiac output is your stroke volume times your heart rate. Can you turn down the volume, please? Thank you. I'm gonna lose my mind today. It's fine. So cardiac output is that stroke volume, the amount of volume of blood that you're putting out from the heart, multiplied by your heart rate. So what is the total volume per minute? Okay, that's your cardiac output basically. So how much of that is gonna pass through the kidneys? Okay. Renal blood flow rate, how much blood is flowing through the kidneys per minute? Okay, so blood flow rate times renal fraction. Renal plasma flow rate, renal blood flow rate times the fraction of blood that is plasma. So obviously if you are dehydrated, that means that you have less plasma, so this renal plasma flow rate would be lower. Okay. Filtration fraction, part of plasma that is filtered into the lumen of Bowman's capsule. So look at this, 19%, okay? About 20%, 1/5 of your plasma is being moved into the glomerulus, or being moved through the glomerulus into the Bowman's capsule and making your filtrate. 20% of your plasma is ending up in the filtrate. Do you think that's going to make a huge difference if you don't reabsorb things? Yeah, it's a huge amount of fluid that you're losing in that initial filtration step. So glomerular filtration rate, how much filtrate is being produced each minute? 180 liters per day. Now, you guys know that I don't make you memorize numbers. However, 180 liters. On average, you're producing 180 liters of filtrate a day, but you are reabsorbing the vast majority of that because your urine production is only going to be between 1 to 2 liters. Okay? Now, you can imagine how many times you go to the bathroom per day, okay? And an estimated volume of how much you're producing. Imagine if you were producing and 180 liters was leaving your body. First of all, do you have 180 liters of fluid in your body? No. So this whole process of creating filtrate and going through reabsorption and secretion is going to end up with a total production of 1 to 2 liters of urine that ends up leaving your body. Okay, so again, you don't have to look at these numbers, but what I think is important to look at is just how much blood is flowing into kidneys, how much of that is plasma, how much of that gets made into filtrate, and how much of that ends up as urine. Okay, so you can see that as we go through these processes, okay, we are reducing that volume that ends up being urine through the process of reabsorption. All right, so our filtration membrane we've already discussed, so make sure that you are familiar with what makes up that filtration membrane. And what is going to be able to pass through that filtration membrane? We said water, ions, some small molecules, small, medium-sized molecules. Large ones are not going to be able to pass through. So some of those larger type of things that can pass through but generally don't would be albumin and some hormonal proteins. Okay, so if a few of those happen to make their way through that filtration membrane, we need those. We need albumin to maintain our blood osmolality. Okay, we need hormones for our homeostatic functions within our body. So those are immediately going to be reabsorbed, and they are not going to be carried out with the urine. So remember I talked about how the only way that you would really see those things in the urine is if there was damage to the filtration membrane, and that would cause a large spillover of those substances into the filtrate, which would be difficult for them to be reabsorbed in their entirety. So filtration pressure, okay, that again, that is what is our driving force for creating filtrate. Okay, so this is the pressure gradient responsible for filtration. And this is what is pushing that fluid from our glomerular capillary out into our Bowman's capsule. So we also have some other types of pressures that are interacting, okay? And this is what is giving us our final number as far as the pressure that's allowing this fluid to pass through, okay? I'll go over these quickly, but it's going to make more sense when we look at this picture, okay? So we have a glomerular capillary pressure, okay? So obviously this is the pressure within that capillary, and this is what is going to be pushing fluid out into the Bowman's capsule. We also have capsule hydrostatic pressure. So this is the pressure being exerted by the filtrate that's already in the lumen, so it's pushing back against that glomerular capillary pressure. Blood colloid osmotic pressure, just like we talked about with albumin, This is the osmotic pressure that is controlling the movement of water. Okay, so again, this is going to act against that glomerular capillary pressure because it's going to want to hold water in the blood. Okay, so here we see a pictorial representation of this. So that big blue arrow right here is our glomerular capillary pressure. Okay, it's a big arrow because it is a large pressure, and that is forcing fluid out from the glomerular capillary bed into our Bowman's capsule. Okay, again, we have that capsular hydrostatic pressure from the filtrate itself that's kind of pushing against that. And we also have our blood colloid osmotic pressure that's trying to hold onto the water and keep it in the blood. So ultimately, if we want to make filtrate, our glomerular capillary pressure has to be greater than the combination of these two pressures acting against it. Okay? If we don't have a high enough glomerular capillary pressure, we won't be able to override these two pressures, and we won't be able to form a filtrate. Okay? So I want you to think about that in terms of blood pressure. If someone has a very low blood pressure, what is that going to do in terms of filtration production? Well, if you don't have a very high blood pressure, how can you force that fluid out? Okay? Because the pressure in the rest of our body is also going to affect the pressure in our glomerulus. On the opposite side of that coin, if we have a very high blood pressure, okay, we're going to increase this glomerular capillary pressure. Okay, these two aren't really affected. If we have a very high blood pressure, okay, then we are going to force more fluid out into the capsule, which means we're going to generate more filtrate. Okay, so that filtration pressure is going to be important for determining the amount of filtrate that we are producing, as well as how fast we are producing it. All right, so an example of a disease that would affect filtration is glomerular nephritis. So anything with the prefix neph or nephro has to do with the kidney, okay? Itis meaning inflammation of. So this is an inflammation specifically of the nephron, okay, of the kidney. And in that nephron, we're specifically having inflammation of the glomerulus, okay? So if we have inflammation, you guys can think back to what we talked about in chapter 22. We have those 5 things that are going to be associated with inflammation. Heat, pain, increased vascular permeability, redness, calor, dolor, tumor, rubor, and loss of function is the last one. So if we have that increased, we have that swelling, that tumor function, due to that increased vascular permeability, then we're going to increase that permeability of those fenestrations, meaning proteins can now enter the filtrate, okay? And that filtrate, because it has those blood proteins that are responsible for creating an osmotic pressure, okay, is going to help draw more water out to follow that concentration gradient, which means that we are going to increase our volume of filtrate, okay? So if we affect the movement of things across that filtration membrane, we are going to affect how much filtrate we are making, as well as what ends up in that filtrate. All right, so we talked about the fact that you can control your glomerular capillary pressure by controlling blood pressure. Okay, so if your systemic blood pressure is too high or too low. But what's really cool about your kidneys is that they have a mechanism for compensating for that and directly controlling the blood that is flowing into and out of that glomerular capillary bed. Okay, so let's go back and look at the structure of the Bowman's capsule again. Remember how we talked about our afferent arteriole and our efferent arteriole? And we talked about the fact that both of these vessels are going to have bands of smooth muscle surrounding them. So if we, oh, go back. If we think about the fact that we have those bands of smooth muscle, those are going to have the ability to vasodilate or vasoconstrict either of those vessels. Okay, so if we alter the diameter of either of those vessels, we are going to alter the blood flow through them. So we remember that resistance, okay, low resistance is going to make things easier to flow. So if you have a low resistance to blood flow in the afferent arteriole, that means it's going to be very easy for blood to flow into that glomerular capillary bed, creating a high glomerular capillary pressure. Okay? If you have a low resistance in the glomerular capillaries, okay, that means that you're also going to have a high glomerular capillary pressure. You can also create a high glomerular pressure by constricting the efferent arteriole and preventing blood from leaving the glomerulus as easily. Okay, so you basically kind of slow down the blood flow leaving the glomerular capillary bed, increasing the pressure behind it in the glomerulus. Okay, so let's look here. So here's our afferent arteriole, here's our efferent arteriole. If you're flowing in easily, if you constrict this, okay, you're going to create a backup of pressure in the glomerular capillary bed itself. All right, so as we continue moving through our vessels, okay, remember how we talked about you're creating this filtrate, you're losing a lot of that plasma volume to creating your filtrate. So as you lose that volume, as you're leaving through the efferent arteriole and you're moving into the peritubular capillary beds, that lower volume is also going to generate a lower blood pressure. So that lower blood pressure is going to continue as we move through those capillaries associated with the rest of nephron. Okay? Excuse me. All right, and again, we can physiologically change the constriction or dilation of the afferent or efferent arteriole to control the pressure that's happening in the glomerular capillary bed, as well as control how much filtrate is being produced as a result. Okay. So how do we do this regulation? Well, we have autoregulation. So there's a myogenic mechanism, myo meaning muscle. So as your systemic blood pressure increases, your afferent arteriole will actually constrict to prevent that systemic blood pressure from affecting the glomerulus and affecting the amount of urine that's being produced. Okay? So we also have tubuloglomerular feedback. So this is an increased rate of flow of filtrate past the cells of the macula densa. You guys remember where the macula densa is? That's in the distal convoluted tubule. Remember, it runs right next to our afferent arteriole, so this is part of our juxtaglomerular apparatus. Okay, so if we have an increased rate of flow past those macula densa cells in the DCT, we send a signal to the juxtaglomerular apparatus, which remember is part of that afferent arteriole. It's part of those bands of smooth muscle that surround it, telling that afferent arteriole to constrict. Okay, so if we're making too much urine, we have too much filtrate that's passing through there, we send a signal back to that afferent arteriole, hey, you guys need to tone it down a notch. Okay, we constrict it, we reduce that blood flow, therefore reducing the rate of filtrate production. Okay, so these are two different ways that we can regulate the glomerular filtration rate. Okay, how much filtrate are we producing in the glomerulus? So those are both auto-regulation, okay? We have no control over them. These are involuntary and they control themselves, but we can also have influence through sympathetic stimulation, specifically through the release of norepinephrine. So we know norepinephrine and, our sympathetic stimulation in general is going to result in vasoconstriction to areas that aren't important for that fight or flight response. So we're going to constrict our small arteries and our afferent arterioles, which means that we're going to decrease renal blood flow, also filtrate formation. So during shock or intense exercise, okay, we are going to have an increased amount of sympathetic stimulation, reducing our rate of filtrate formation, okay? I want you to think about this for a little bit too, okay? We know that one of the things that happens during sympathetic stimulation is we increase blood pressure. So by increasing blood pressure, we would already have these autoregulatory mechanisms in place. But do you really want to stop to go to the bathroom because you've produced a ton of filtrate and eventually urine? Do you want to have to do that while you're fighting a bear or running away from a bear? No. So that sympathetic stimulation is going to shut that process down so that it is not going to continue to affect you under that sympathetic type of environment. Oops. Okay. All right, so I think we'll be able to get through tubular reabsorption today. How many slides? We got 59 slides. All right, so tubular reabsorption is our next stage in urine production. And this is where we are picking up important things and putting them back into the blood from that initial filtrate that we had formed. So there are different ways that we can pick those things back up. Diffusion, okay? Remember, this is a passive process. Things are just diffusing from one side of the membrane to the other, and typically this is associated with some kind of concentration gradient. Facilitated diffusion, so that diffusion gets a little bit of help. Active transport, remember active transport, active because it requires ATP, you need energy. Typically this is associated with moving things against a concentration gradient. Symport, so we're moving two things at once. So for example, we typically, and we saw this with the digestive system, when we're moving glucose, a lot of times we also move sodium at the same time. And then osmosis, okay, is that movement of water. So as these substances are picked back up from the filtrate, they're transported across that tubular epithelia and then into the interstitial fluid, and then they are moved into the blood capillary to return to circulation. So our proximal convoluted tubule, you guys should be familiar with the basic structural aspects of a endothelium. The apical surface is what is facing the lumen, okay? That's typically like the top of the cell. Our basal surface is the bottom of the cell, and this is typically going to be on the outside of the tube and what is facing the interstitial fluid. And then our lateral surfaces are on each side of our endothelial cells, and they are very tightly attached to one another to prevent the movement of things from between the cells and into the interstitial fluid. All right, so you'll notice as we continue to go through the rest of the tubular segments, we're going to see a lot of information about what types of ions are being moved at these different locations, okay? So as you guys are trying to reference what is being moved, you can look at slide 45. No, wrong one. Aha. Okay, well, 45 and 46. Along with 48, so like this whole stretch of slides right here, okay? And they are going to tell you what types of substances, okay, what important things are being moved out at each segment, okay? So that being said, look at this as a reference as we are discussing these different things that are moving, okay? So sodium is going to be actively transported across the basal membrane, okay, into the interstitial fluid. And remember what we talked about with moving things across a membrane? If you are moving a solute from one place to another, okay, typically you are going to decrease the concentration as you're moving solutes out. So for example, in the proximal convoluted tubule, you have your solutes, all your ions plus water. As you move those solutes out, you make that filtrate less concentrated, which means that water is going to want to do what? It's going to want to follow the solutes, okay? And so you're also going to have movement of water along with the movement of solutes. So the water movement is going to be via osmosis, okay? And depending on the ion, we may have active transport, diffusion, things like that, okay? But for sodium, it's active transport across that basal membrane. And I want you guys to visualize the cell real quick, okay? Yeah, you can't see, okay. Okay, so this is our apical surface, which means that our lumen is right here. This is our basal surface. Can you see around my head? Okay, and then on each side we would have our lateral surfaces. So if we are actively transporting sodium, my favorite color for sodium is blue. I don't know why. If we are actively moving sodium out of the cell into the interstitial fluid, okay, across that basal membrane, then that means that we are going to have create a concentration gradient. So as we actively move that sodium out, it's going to draw more sodium into the cell itself, okay? So that's what it's talking about in this next part. Because of active transport, the concentration of sodium is low inside of the cell, and so sodium is going to move from the filtrate into the cell, okay, through this apical membrane, and then it'll be actively transported out into the interstitial fluid, okay? So passively, it's going to move from the lumen across the apical surface, and then active transport out of the cell, okay? So we create a gradient here, and it's going to draw that movement. All right, so other things that are going to be transported across the apical membrane include chloride, glucose, amino acids, and water. Now think about that for a second, okay? Most of these, the addition of potassium, are also going to be transported through the basal membrane, which means that all of these substances are going to end up back in our blood circulation. Why is it just these substances that are primarily being absorbed here? In the very first segment of the nephron, after we've made our filtrate, okay, we want to pick things back up that are the most important to our basic functioning, okay? So sodium and chloride, okay, both of those are going to be very important for our nerve function as well as our muscular function, okay? As well as they're just ions that we need for a lot of different functions within the body. Glucose, okay? Glucose is obviously extremely important, and we need that in our blood to make sure that's available to our cells, okay? Same thing with amino acids. Those are incredible building blocks that we need for making proteins and other things, okay? So when you think of the proximal convoluted tubule and you think of reabsorption, I want you to think of what types of things are most important for my body to use, okay? So again, those ions, sodium and chloride, glucose, amino acids, and then also because we have created this gradient by moving ions across, we're also going to be moving lots of water. Okay? So again, we need to reabsorb that water because we just lost 1/5 of our plasma volume, okay, in forming this initial filtrate. So we need to make sure that we pick that up so that we can maintain our blood pressure and return that water to our plasma. All right, so here is a nice pictorial representation. Here is our lumen with our filtrate. Here is us moving our different substances across, and then it shows you what type of transport is happening on the basal surface to allow for those ions and molecules to be able to move back into blood circulation. Okay, so if you needed a better way of understanding what I drew up here, you can look at this slide. All right, so reabsorption in the proximal convoluted tubule continued. So depending on what substances we're trying to move, okay, if they need a carrier molecule, our movement of those substances is going to be limited. Okay, so you can only move as many things as you have carrier molecules. Excuse me. This is what, like over a month now that I've had this cough and it just will not go away. Okay, so we just talked about the fact that glucose is one of the things that's picked up, because we need it in our body. We need it for our cells, we need it for making ATP. In conditions like diabetes mellitus, where you have an excessive amount of glucose in the blood, you're not going to be able to pick up all the glucose that has moved out into the filtrate. Okay? Do you guys ever see, I'm sure that none of you have ever watched a full episode of I Love Lucy, but I'm sure that you've seen little video clips online of the episode, and you should look this up. I'll post a link for you, where Lucy and Ethel get a job at a chocolate factory. Okay, and in the process of working in the chocolate factory, they are supposed to pick things off the line and do something with them. Okay, well, the speed of the line keeps moving faster and faster, and then there's more and more chocolates, and they can't pick them up fast enough. Okay, so hilarity ensues, as I'm sure you can imagine, but I want you to think of that in terms of chocolate is glucose. Okay, the more glucose that you have in your blood, the less that you're going to be able to pick up as that glucose in the filtrate is moving through. Okay, so if that rate of if the amount of filtrate of glucose in the filtrate exceeds the rate of reuptake, then we are going to have a spillover of glucose into our urine that is being produced. Okay, and I think that we had discussed this before. But that is why one of the ways to tell if someone's blood glucose is controlled, or to tell if someone has diabetes mellitus, is to test the urine for glucose. Okay? So the reason for glucose in the urine is because there's so much in the blood plasma, there's so much that's being spilled into the filtrate that we just can't pick it back up. Okay. All right, so the other important thing that is happening here is that our filtrate volume is reduced by 65% due to that movement of water through osmosis. Okay, so almost all of our water, okay, that is going to be reabsorbed is going to be reabsorbed at the level of the proximal convoluted tubule. Okay, so what I would encourage you to do as we're going through here, if you're not already doing this, is to make yourself a list of different segments of the nephron. Okay, so, and we'll start with PCT because that's what we're talking about. Okay, you want to say which of the 3 stages of urine formation is occurring here? Well, reabsorption. pardon my sloppy handwriting. Okay, what are we reabsorbing? Okay, we're reabsorbing sodium, chloride. Okay, those are our primary ions. We're reabsorbing glucose and amino acids. Okay, our nutrients and building blocks. And because we are reabsorbing all of these solutes, we are going to reabsorb lots and lots of water. Okay? So this is a good way to start with your organization of what's happening in each segment of the nephron. Okay? All right. So moving on to reabsorption in the loop of Henle. Yeah, you guys are going to get wrecked on the next lecture. It's fine. So we're going to continue reabsorption in the loop of Henle. And remember how we talked about the changes in the epithelium as we move through that tubing system? Okay, that's going to alter what we are reabsorbing in this loop. Okay, so spoiler alert, as we move through the loop of Henle, we are going to absorb different things. Okay, so as that loop of Henle descends into the medulla, okay, it's important to note that the medulla, the interstitial fluid that's in the medulla, becomes increasingly concentrated the deeper you go. Okay, so let's skip ahead to look at this diagram. So here is representing the cortex going down into the medulla. So as we extend this loop of Henle down into the medulla, you can see that the concentration of solutes increases as we go down into that loop of Henle. Okay, so as we descend, we are increasing the concentration of solutes in that interstitial fluid, and as we ascend, okay, we are going back up to areas that are less concentrated. Okay. So the descending segment, okay, also notice that there are thin segments and thick segments. We haven't discussed this yet. I guess I gotta skip back all the way down here. Okay, this area right here is the thick segment. Here's another thick segment, and then this thin segment is this area that's thinner right here. Okay, so that's what we're talking about. So the descending thin segment is highly permeable to water and moderately permeable to urea, sodium, and most other ions. Okay, so water is moving out of the nephron and solutes would be moving in. Okay, so as we descend into the loop of Henle, our filtrate volume is reduced by another 15% because we are moving out water. Okay, so if you are doing a similar thing like we did with the proximal convoluted tubule for the loop of Henle, you're going to want to divide it into the descending segment and the ascending segment. Okay, so the ascending thin segment is not permeable to water, so we're not moving water at all, but it is permeable to solutes. Okay, so solutes are what we're going to be moving on the ascending side. Now, let's go back to this picture because I really do like this picture a lot. Wink, wink. And if you think about this logically, it makes sense as to why we are moving water on one side and we're moving solutes on the other side. Okay? Remember, the movement of water, okay, osmosis, is driven by concentration gradient, specifically by the concentration of solutes. Okay, so as we are moving filtrate down this descending limb, okay, we're increasing the concentration of solutes in that interstitial fluid, So as we increase that concentration of solutes, that means that more and more water is going to be drawn out as we make our way down, okay? And we are going to continue to move water out until we have equalized the concentration between the filtrate that's in the lumen of the tube and the concentration of the solutes in that interstitial fluid, okay? So we're going to keep moving water out until we have reached an equilibrium, okay? So the concentration is going to be equal at the tip of the loop of Henle, okay, to that interstitial fluid. All right, and we're going to continue to equalize as we move down. So you can see here it's 800, it's 800. But as we descend, remember that concentration increases. So we're going to continue to move water out to keep equalizing those concentrations. So the area where the filtrate is the most concentrated, because we just moved out all this water and we have tons of solutes in it, is going to be here at the loop of Henle, okay? At the tip of the loop of Henle. Now, as we go back up the ascending side, okay, we're not moving water out because we are decreasing our concentration. So how do we equalize our concentrations between the filtrate and the interstitial fluid as we go back up? Well, if we can't move water, the only other thing that we can do is to move solutes out, okay? And as we move solutes out, okay, we decrease the concentration of our filtrate moving back up this ascending limb of the loop of Henle, okay? So this is another image on 45 showing you that movement of fluids through the loop of Henle in the filtrate, as well as what we're moving out, okay, excuse me, at each area of the loop of Henle. All right, so of course, we're not just moving our substances into the interstitial fluid, we're also moving those substances into our capillaries. And remember, the capillary that is surrounding the loop of Henle is going to be the vasa recta, okay? The peritubular capillary beds are surrounding our PCT and DCT, and then the vasa recta is surrounding this loop of Henle. So that's where we are moving either our water or our solutes into. Okay, so again, that ascending limb, not permeable to water, we're moving solutes, okay, into that interstitial fluid and then into our blood capillary. So if I was to ask you a question about where filtrate is the most concentrated through the length of the nephron, like we just said, the very tip of the loop of Henle, okay, the bottom of the loop of Henle, the deepest part of the loop of Henle that has extended into the renal pyramid in the medulla, that tip is the area where filtrate is going to be the most concentrated, because in the descending limb we moved all the water out, increasing that concentration of solutes, okay, and by the time we start going back up, we're moving out solutes, so we are decreasing our concentration as we move back up, okay? But the area where it is the most concentrated is at the bottom or the tip of the loop of Henle. All right, so again, more diagrams showing you these movements of water, okay, or ions. And then we will start next week on discussing this reabsorption in the distal parts of the nephron. So again, if you guys have questions, let me know. I appreciate you being patient with chaos today. And I will be online at least until like 3:30. So if you guys need to get a hold of me, you can just message me in Microsoft Teams. All right, have a great weekend, guys. I hope you do something to help your sanity. I'm going to go outside and play with my chickens, and that is going to make me feel a lot better. So play with your cats, your dogs, whatever animal you have around you, or just go outside and watch the birds. That's always fun. So I will see you guys next week, and thanks for paying attention. Bye.